All right, in this video we're going to talk about the various stages of mitosis. And there are basically 5 stages that we'll talk about. The first stage is prophase. So remember, mitosis is division of the nucleus. So one of the first things that happens during prophase is the chromatin condenses into visible chromatins. Remember, the DNA has already been copied, so the chromosomes are made up of 2 sister chromatids. In animal cells, the centrioles are going to move to opposite poles and begin forming the spindle fiber, or the spindle apparatus, I should say. Protein fibers cross the cell to form the mitotic spindle, or spindle apparatus. These are made up of microtubules, actin, and myosin, the same stuff that makes up the cytoskeleton. And it coordinates the movement of the chromosome. They will attach to the centromere of the chromosomes and move the chromosomes through the various stages of mitosis. The nucleolus is disappearing. The nuclear envelope or nuclear membrane is also breaking down. The transition to metaphase, or what we call prometaphase, is a term that's probably new. You probably didn't talk about this in freshman biology. This is when the spindle fiber is actually attached to the centromeres. This creates what we call kinetochores, where you have the microtubules crossing across the cell. So the kinetochores connect the centrioles to the centromeres. Again, centrioles in animal cells. And the chromosomes are going to start moving around the cell, but they're not going to line up yet. In metaphase, chromosomes are going to line along the midline or the center line of the cell. It's called the metaphase plate. Again, the spindle fibers are going to be what controls this movement and lines them up. Again, by lining them up, it makes it easier and more likely that each of the daughter cells will have 1 copy of each chromosome. Anaphase, the third phase, the sister chromatids separate at the kinetochore. They're going to begin moving to opposite poles, again through the help of the spindle apparatus. And they're basically motor proteins that kind of walk them along the microtubule. Again, the proteins are actin and myosin, the same proteins that make up our skeletal muscle. So since we're using energy here, we need to increase the production of ATP by the mitochondria. The poles themselves are actually going to move further apart, pulling the chromosomes, sister chromatids apart. Here we can see that. So there are proteins that would normally hold the sister chromatids together, but they are inactivated, which allows them then to separate. So here we have 1 chromosome with 2 chromatids. And then in anaphase we have 2 chromosomes, single strand. So again this just kind of talks about how the chromosomes separate during anaphase. And then finally in telophase, the chromosomes are going to be arriving at the opposite poles of the cell. The daughter nuclei are going to begin to form around each set of chromosomes. The nucleolus is going to reform around each set of chromosomes. The chromosomes are going to disperse and they're going to become uncondensed into that chromatin state. Spindle fibers are going to disperse, the spindle apparatus is going to break down. And then normally cytokinesis, or the splitting of the cell, is going to follow this process. So cytokinesis is the actual splitting of 1 cell into 2 cells. In animal cells, there's a constriction belt that kind of forms around the center made of actin filaments that kind of pinches the cells in and eventually pinches them off into two individual cells. That pinching in forms what's called the cleavage furrow, and the cell splits in two. Here we can see the stages during here in an animal cell. This is a whitefish blastula. This is in the developing embryo of a whitefish. Another picture here in animal cells. In plant cells, because of the cell wall, cytokinesis is a little different. So there's a cell plate that forms between the two sets of chromosomes, the two nuclei. Vesicles line up at the equator. These vesicles come from the Golgi apparatus. The vesicles fuse to form two cell membranes. So you got one on each side of the cell plate. A new cell wall is then laid down between the two membranes at the cell plate. New cell wall is going to fuse with the existing cell wall, and what you end up with is two cells about the same size. So here you can see kind of the vesicles and the cell plate there. Here we see mitosis in a plant cell. Here we can see the different stages in onion root tips, which are growing. You can see the different phases.